Very warm welcome. Now, the second type of mutagen, which is chemical mutagen, is the broadest mutagen that incorporates so many chemicals which are capable of using mutation, and these chemicals are available freely in the nature as well as man-made chemicals are capable of inducing chemical mutagenicity. Ultimately, the effects are visible or organism. Among this category, subgroup of chemical as per their effect have been classified. They are called as base analogs. They are alkylating agents, intercalating agents, and adduct forming agents. In the group of base analog, these are chemicals which are mutagenic in nature, and these chemicals have these chemical compounds have capability of base substitution. They can substitute purine by purine or pyrimidine by pyrimidine during the replication. Among them, there are potent examples. One is 5-bromouracil, and second is your 2-aminopurine. 5-bromouracil is a uracil derivative, and it behaves as adenine in their keto form, as we can see in structure, and they start pairing with A. In the rare enolic form, the 5-bromouracil pairs with 1A, and such unpairing results in transition mutation. These transition mutations are basically resulting into mispairing of bases of nucleotide during the replication, and such mispairing give rise of point mutation. The other molecule, which is your aminopurine, is adenine, also adenine analog. Usually, it starts pairing with T when incorporated due to the replication process, and sometimes in the rare cases, it also pairs with 1A. So consequently, in next replication cycle, give rise of your transition mutation, and ultimately seen as point mutation. These analogs are creating changes as mispairing during the replication. The second group of chemical used for induced mutation are called as alkylating agent. The example of alkylating agents are mustard gas, was used in World War I. In the World War I was applied in Vietnam. It was very poisonous gas. And another example is EMS, ethyl methane sulfonate. These alkylating agents donate an alkyl group to amino group or keto group present on purine. These, alkyl group may be methyl or ethyl. When they, alkylate the keto group, they, get attached to the sixth position of guanine and number four position of thymine. The keto group present in the bases. The EMS alkylates keto group of number six position of guanine. The alkylation leads to alteration in base pairing affinity of these nucleotides, and it leads to transition because the affinity gets changed, and this changed affinity causes the transition, such as we can see ethyl methyl sulfonate when reacts with alkylating agent or reacts with the guanine, it forms the 6-ethyl guanine, and it starts behaving like an analog of A, and it starts pairing with T. Earlier, guanine was pairing with C, G was pairing with C. So 3 bond gets altered with bond, and another one purine got replaced with another purine. G got replaced with T, and consequently C got replaced with A. So it gives rise of transition patient. So alkylating agents are also develop transition patient, and this transition leads to change in the mispairing, and ultimately results in your point mutation. The third group of chemical which are used in induced mutation are basically intercalating agent. These intercalating agents, as the name suggests, intercalating. They are present between two. So these are the chemical structure, chemical molecule having dimension and shape that allows them to wedge between the nucleotide of DNA. So they get inserted in between the two nucleotides because of their flat structure, and such insertion leads to development of distortion in DNA, and it develops, it becomes visible as a kinky structure, and it leads to unwinding of DNA. The unwinding effect is not good. It leads to formation of loop in the double strand DNA, and that double strand DNA loop may cause deletion, because during the replication, what happens? The loop portion is not copied by the replicating machinery, and they get deleted. Such sequences get deleted, and deletion results in frame shift mutation. One of the example of this intercalating agent is Etherium bromide. Etherium bromide is a fluorescent dye. It is used in DNA stain. So with the help of this stain, we can visualize DNA after our experiments, which are carried in your gel electrophoresis. Usually, due to not the proper management of these Etherium bromide, they are discarded carelessly, and these Etherium bromide to the ecosystem and absorbed by downstream organisms. Such absorption causes access of such molecule in the DNA of the recipient organism, and it leads to development of changes in their genetic makeup. Some other examples are roflavin, And there are several intercalating agents which have been used as medicine to treat several cancers disease because they stop the replication of the DNA caused defects in them. There are some example like one example is doxorubicin. Doxorubicin. It is used as an anti-cancerous agent and it is used for treating Hodgkin lymph. The another one is your dactinomycin. It is used for treating the sarcomas. So intercalating agents are the chemicals which are harmful in their natural condition, but used as a using the mutation, or if they are taken naturally, they causes the induction of mutation. Their knowledge has resulted in application, and they have been used as a medicine for treating the several uh, disease like cancer.